Okay, so thank you for the highlights for the ERGA committee. So we'll go on with the talk of the month. So it is a pleasure for us to have talk Oosterhout with us, who will be presented by Camilla. Mindful of the time, we will try to stick on 30 minutes max talk and see if we have some time for questions. Camilla.
Very nice, thanks, Claudio. So it's my great pleasure here to introduce you to Kok van Oosterhout. So we are continuing with the presentations that are connected to the data analysis committees. So our idea, as we have been approached by different people already, leaders in their fields who want to collaborate with ERGA, who want to form groups and analyze genomes under different perspectives together with ERGA. So we are thinking and considering to form new working groups under different topics. And so the talk today is going to illustrate one of these possibilities so that we can continue with this discussion afterwards next year within the data analysis committee, and of course always including anyone from the members who are interested in participating in forming this potential or one potential new working group. So I'm going to introduce here the background from Kok, because he has quite a long history already in science, starting with his PhD in the Leiden University, working on captive metapopulations of butterflies, and particularly on the effects of inbreeding on fitness components and genetic load of Bicyclus aninana. He then moved to the University of Hull as a postdoctoral fellow and later obtained a lecturer position working on conservation genetics, captive breeding, and reintroduction using the rainbow fish Poecida reticulata, known also as guppy, as empirical model. Guppy, okay. Many of us are acquainted with the number of widely used software applications developed by Kok, including microchecker and hybrid checker. And he now works on evolution and population genetics, and particularly on hybridization, progression, evolution of immune and resistance genes, and host parasite coevolution of a wide range of species. He's professor of evolutionary genetics at the University of East Anglia and acts as a deputy director of the Earth and Life Systems Alliance. And today, Kok is going to give us a talk on assessing genomic erosion in threatened species. So Kok, the floor is yours. Welcome to the plenary today.
Thank you very much, Camilla, for a kind introduction, and welcome everybody. Really nice to be a part of this group and present some of our research. So let me share my screen with you. So indeed, what I will be talking about today is assessing genomic erosion in threatened species. And the main motivation really of this research was that being part of the IUCN Conservation Genetics Specialist Group and seeing how species that have undergone bottlenecks, how they are sometimes being up or down listed in the red list, how they still can suffer from genomic erosion. So the red list reports a lot of data on almost 150,000 species, the direct threats to species survival, like habitat loss, invasive species, and so on. It's a beautiful, beautiful resource, and if you haven't looked at it yet, I mean, I really recommend you having a look at it. You can register, and you can then basically examine the red list, looking at all kind of threat categories for these 150,000 species. For example, here I tick the boxes critically endangered, endangered and vulnerable. So these are the threatened species. So there are 141,500 or so threatened species out of 150,000. But you can do this for any group of species, or you can look at population trends, or at the specific threats or particular habitats. Really a brilliant resource of information. So these IUCN red list data, they really report on the present threats to populations, what causes population demography and population size and population habitat fragmentation. So these direct threats also have long-term consequences. We know that, of course, from population genetic theory and evolutionary theory. For example, historic bottlenecks lead to inbreeding and drift. Despite species rebounding from these population bottlenecks, sometimes quite remarkably so, like the Mauritius kestrel and echoparakeet, pink pigeon, they do leave a signature of in the genome. Basically, this bottleneck erodes, of course, variation, and that is something that we can analyze now with whole genome sequence analysis. So what I will be talking about is really kind of this imprint on the genome caused by kind of these direct threats to populations, and I will refer to that as genomic erosion. Now, to be a bit more specific, so genomic erosion, I list kind of 4 different threats. First and foremost, and everybody is very much aware of that, that's of course the loss of diversity. Genetic diversity that has been investigated since, well, the early 1970s, really. Brilliant work started with Crow and a lot of people, Franken, and a lot of people working on genetic diversity loss, initially with allozymes and then later, of course, DNA techniques. So that has really been the kind of the paradigm in conservation genetics. However, also during genomic erosion, a lot of things happen to the genetic load, and I will explain a little bit more about that in the subsequent slide. There's also a severe problem with maladaptation due to rapidly changing environmental conditions and maybe a lack of adaptive evolutionary potential of the populations, because the diversity has declined. Heritable variation also can go down. There's maybe issues with domestication, particularly in zoo animals or animals that are reliant on food supplementations in the free living environment. You get maladaptation or adaptations that are maybe not as good, for example, if those animals are reintroduced into the wild. And then there's a potential problem with genetic introgression after hybridization. Again, this happens naturally in a lot of species. A lot of species have had some a history of hybridization in the past. But of course, right now, due to range shifts and habitat and environmental change, also mediated, human-mediated transport, the incidence of this genetic introgression is increasing. And we can, of course, identify those signatures in the genome. So I will be focusing particularly about the mutation load, which I think has taken a little bit of a backseat in the whole discussion about species conservation and conservation genetics. So what is genetic load really? Well, a genetic load is a consequence really, or the problems come from genetic drift and inbreeding. Those processes expose bad mutations, recessive mutations in homozygote conditions. Now, most species, most outbreeding species have a lot of bad mutations in their genome. They are often in quite low frequencies in populations, and they tend to be recessive. And they are recessive, which means that they're not expressed if they are in heterozygote condition, or they're only partially expressed in heterozygote condition. And you can understand that because if you have a dominant variant that is bad, well, selection can observe it straight away, and selection would remove that variant very quickly from the gene pool. So as a consequence of that, you have a lot of recessive deleterious mutations in the genome. Now, when 2 copies of the same bad mutations come together, you get inbreeding, and these are in these homozygotes, you get inbreeding depression. Now, if I ask my students what is the effects of inbreeding on the genetic load, then I get various answers. So maybe I would like to ask you, think about what do you think happens with the genetic load during inbreeding? Is it going up? Is it going down? Or does it stay the same? So have a quick think about it. What do you think has the theory and the literature taught you about the genetic load and in relation to inbreeding? Now, very briefly, and not many equations, but something I think which is quite useful to know is what the genetic load is defined as in the population genetic definition. So this is the original definition from Crow in the 1970s. This is really how he and also others before him, Muller and Haldane, how they defined the genetic load. It's simply the sum of the selection coefficients of all the deleterious mutations in the genome across all those loci multiplied with their frequency in the population. So in a formula, it's the sum over all loci, that's the L, Q, that's the frequency, and S is the selection coefficient. So for example, if you had now 1,000 loci with mutations, each with a frequency of 10%, 0.1, and a selection coefficient of 2%, 0.02, then you can calculate the genetic load, and that would be 1,000 times 0.1 times 0.02. That would be 2 lethal equivalents. And lethal equivalence is kind of the unit of the genetic load. Now, it doesn't mean that an individual with 2 lethal equivalents will die. In fact, the genetic load itself doesn't say anything about fitness, because it's independent of genotypes. It doesn't say anything whether this population is fit or unfit. For example, if all these individuals would be heterozygous, well, these individuals will be perfectly fit, particularly if those mutations are completely recessive. Also, when you look at this formula, you can see it's not affected by inbreeding because it doesn't say anything about the actual allele, actual genotype frequencies. It's just the sum of bad mutations in the population. That is all what the genetic load is in the original population genetic definition. Now, that might surprise you a little bit, and often it's confused with the realized load. So what's the realized load? Now, I'm here talking about an individual's perspective. So the realized load is simply the sum of the selection coefficient of all homozygous loci plus the sum of the selection coefficients of all the heterozygous loci multiplied by that dominance coefficient, the dominance coefficient H. And in formula, you can see this sum of S of all the homozygous loci plus the sum of L, the heterozygous loci. And then you have the H and the S for the dominance and the selection coefficient. So basically, again, it's a sum of selection coefficients and the dominance coefficient in case of heterozygotes. Now, in this formula, you can actually see that the realized load does affect fitness because, for example, inbreeding will increase the level of homozygosity and therefore reduce also heterozygosity. And because H is typically very close to 0, you get more of these bad mutations fully expressed in the homozygote condition. So you can see that this is actually a term that is affected by inbreeding. Now, how does it then affect fitness? Well, then you have to get to this, the next section. Suggest you have again that same example of a 1,000 loci with mutations with a selection coefficient of 0.02 and a frequency of 0.1. And then the genetic load, as we saw earlier, is 1,000 times 0.02 times 0.01 is 2 lethal equivalents. Now, suggested this population goes through severe inbreeding, so extreme inbreeding that fixes all those loci for homozygote states. Now, if it's completely drift, there's no selection, then basically the chance of these mutations to fix in their kind of deleterious state, in their bad mutation, is the same as their allele frequency, the 10%. So basically, that's the 10% of all the loci become fixed for the mutant allele, and 90% will be fixing for the wild type allele if there's no selection. And that's important, is pure only drift. Now, so 10% of 1,000 loci becomes 100 loci. They are fixed for these mutations of the selection coefficient of 0.02. And then you get this realized load, and it's only the homozygotes term here, is the sum over 100 loci with each one a selection coefficient of 0.02. So that becomes that 2 lethal equivalents. And now you see that the genetic load and the realized load are exactly the same in this case. And so that's because inbreeding has fixed all these bad mutations, or well, no, actually proportional to their frequency in about 100 loci, and that causes that 2 lethal equivalents become expressed. Now, how does that now affect fitness? And this is basically the original Crow formula. The fitness is 1 minus the selection coefficient, and that is the 0.02, the 2% reduction in fitness, and that is multiplied over 100 loci. So we assume that all these loci have fitness effects that work multiplicatively across all those loci. So 1 minus 0.02 to the power 100 is the same, and that's that term e to the power minus 2. And that minus 2 is basically 0.02 times 100. So that is basically, if you have a small term, 1 minus a small term to a power, then that is the same as e to the power, that small term times the power. So that's 0.02 times 100 equals minus 2. So this is now the actual fitness of that individual, and that is basically 13.5% or so after complete inbreeding. So that is basically the average fitness of an individual in a population with a load of 2 lethal equivalents. And you can also see that the fitness actually is e to the power minus the realized load. So that is how a genetic load and the realized load are affected by inbreeding and how it affects fitness. Now, in very simple graph form, so over here you can see that the genetic load actually doesn't change with inbreeding coefficient, just what we saw from the formula. There's no effect of inbreeding on the genetic load because it's not dependent on the genotype frequency. Now, the realized load increases, and indeed with maximum inbreeding, of an inbreeding coefficient of 1, it becomes the same as the realized load. At the same time, the mass load, that's basically everything that's not expressed, not realized, goes down in a similar fashion as the realized load. Now, you might say, well, listen, I thought that with inbreeding, you also get some purging, and that reduces the genetic load. Yes, that is indeed right. And indeed, you can draw this line now slightly lower down. So basically, the number of lethal equivalents, so the genetic load, will reduce with inbreeding because selection purges some of these bad mutations. And that is because the realized load, once they become realized, they become seen by selection, and selection allows is then able to remove some of those bad mutations. And that's basically purging, but it's also the same as inbreeding depression. So that is actually that fitness loss. So over here, I have in the bottom line, I have now actually the genetic load definition, also used by Crow sometimes, to show that it's the difference between the individual with a genetic load compared to a perfect individual. And that's another definition of the genetic load that you sometimes see in the literature, actually quite commonly see in the literature. So that says, okay, the genetic load increases with inbreeding, but that's because the fitness goes down, and that is inbreeding depression. So here you can see the confusion in the literature. Some people might say, okay, genetic load goes down with inbreeding because of purging. Others may say, well, genetic load goes up with inbreeding because the fitness becomes less, and therefore you have inbreeding depression. So therefore you have less fitness, higher genetic load. And in fact, if you look at the population genetic formula, you say, well, actually nothing happens to the genetic load because it's independent of any genotype frequencies. So whatever you had answered to that question, you will be right. But of course, that's not very, very precise. And therefore, we wrote this paper in Nature Review Genetics. So this is together with Giorgio, and I saw also that Meerte was on the call. So this is a kind of a review about the genetic load, trying to explain the population genetics, the classic population genetics, and some really modern techniques to assess it at a molecular level. So next part of the talk is about the pink pigeon, and here I'm going to talk about how we've implemented all this knowledge into conservation action, say management plan, and conservation actions to save the pink pigeon. So it's paper. This is the paper actually which I will be discussing, which we published earlier this year. So pink pigeon is a real conservation success story. It has been downlisted twice on the IUCN Red List. It was critically endangered in the early 1990s, and it went down to endangered. And recently in 2018, it became downlisted to vulnerable. So what we studied was the genetic variation, the genetic load, and also the long-term viability using computer simulations. So in this study, which started actually quite a few years ago, we still used red sequencing of 175 birds. We also did the whole genome sequencing of six birds, and we also looked at the fitness of birds in pedigrees and also in the wild. So what's happened to the pink pigeon? Well, the population went through a severe bottleneck of about 12 or so individuals in the late 1980s, and it then came back. So over here you can see the demography, population size. It came back in the early 1990s, and around 2000 or so, it reached a carrying capacity of about 400 to 600 individuals in the free-living population. Now, this remarkable rebound was really facilitated by reintroductions and also from UK zoos and a lot of conservation actions in Mauritius, and really quite a concerted effort by a lot of people. So, however, what we see is that the genetic load may actually now hindering further recovery, and we wanted to investigate that further. So one of the things we saw is that in the even during the population recovery, which was say in the 1990s. So here we have on the x-axis the time, say starting from 1994 to 2007. Then and on the y-axis the observed heterozygosity. What we see here is that there is a reduction in observed heterozygosity, even though in the population demography rebounded. So basically the population size increased. Over here, it's actually in this period that we sampled. So from here to here. And in this period that the population goes up in size, we do see a remarkable reduction in observed homozygosity or heterozygosity in the genome of these birds using red sequencing. So strong genetic drift. You can also analyze this by comparing directly parents and offspring. All these birds are ringed, so you know exactly the parents and the offspring, and we have blood samples of those birds, and we calculated their heterozygosity. And from that, we saw that the heterozygosity actually reduced markedly in one generation. Using this data, you can also use this formula to calculate the effective population size, and that's nowhere near the 400 birds that we have in Mauritius. It's around, well, actually, it's less than 10 birds, about 7.6 birds. So it's an incredible amount of inbreeding, despite the fact that there are so many birds on the wing now in Mauritius. So not such a good story, really, from a genetic point of view. Now, this also leads to very clear signatures. Here we see these runs of homozygosity that you probably all have seen, large areas in the genome. So this is chromosome 1, chromosome 2, etc. Large areas in the genome are completely homozygous. This is the least inbred bird, and these are the two most inbred birds. And if you calculate the actual inbreeding based on these runs of homozygosity, well, that's an inbreeding coefficient significantly more than a brother-sister mating. So it's really a lot of inbreeding going on in these birds, causing loss of diversity and increase of homozygosity. Now, and you can imagine, of course, if you have a mutation in such a region, and that is recessive and deleterious, it will become expressed. So there's a lot of convergence of this kind of this masked load into this realized load. Just what I explained, inbreeding increases the amount of realized load. Now over here, I will show you what we've done subsequently with a nice computer simulation by Hernan Morales and Alessandro Pinto. So basically, this is a computer simulation where we have simulated the habitat in Mauritius. So what you see here in green is the forested habitat of Mauritius. So this is the island of Mauritius. And what you can see is that the amount of green is slowly declining. What you also see is the number of red and yellow points, and that are pink pigeons. So this is a computer simulation done in SLIM. And each individual is parameterized, has a whole chromosome simulated. So this was run on a computer cluster for several weeks. And basically, we have simulated the genetic load and the amount of inbreeding that you expect due to habitat decline over time. Now, what you can see is that, of course, as habitat declines, the population becomes more and more fragmented, and this also increases the inbreeding coefficient. So over here on this graph, you can see the forest cover, and here from 1773 down to 2000 or so, you see this is the forest cover, which is documented. It's actually very well documented in Mauritius how the forest has declined over time. So we have really good data for that, historic data. And of course, this is the kind of the simulated population size of pink pigeons. And you see indeed that echoes very much the habitat degradation and amount of forest cover that is left. What you can also see is that the inbreeding coefficient goes up. But what you perhaps see that actually the inbreeding coefficient only goes up very, very slowly. It takes quite a while for inbreeding to really go up. Similarly, the heterozygosity, well, it only starts to decline markedly around 1950. And in 1950, well, the habitat would have been, well, very much reduced. It would maybe be something around this size. So you would have a lot of habitat loss already, a lot of habitat fragmentation, but the amount of heterozygosity would still be very similar to that of the ancestral population. And only more recently, we see that decline. Now, what you also see is that the genetic load is increasing, and the increase is even more delayed than the loss of heterozygosity. So we can really see that this red bar, which is the realized load, goes up, particularly, say, in more recent times, around 2000, right when the population actually was recovered. We see that there is a marked increase in the realized load, which causes loss of fitness and inbreeding depression. So now, the next step that we want to do is we want to really get to grips with actually identifying the genetic load at a molecular level. And this is work that has been done by Sam Speak, a PhD student in my group. He uses a bioinformatics method based on work in human genetics, CAT scores. And what he has been studying here is the CAT scores at ultra-conserved elements, so areas in the genome that are basically really conserved across the whole phylogeny. There are very similar, for example, between birds and mammals and even fish. You can see the same kind of DNA sequences in these ultra-conserved elements. Now, he's using those ultra-conserved elements for two reasons. First of all, the level of conservation is thought to have arisen because there's strong purifying selection. So basically, mutations in those areas are not allowed. They are basically selected against. So anything that does is mutated is probably quite bad. And secondly, UCEs are very nice to compare because you can align them and you can use also CAT scores and say basically scores that quantify the impact of mutations from model organisms to your non-model organism, which in this case is a pink pigeon. So in that sense, you can assess the fitness impacts of those deleterious mutations for individuals. Now, over here you see the chromosomes, the genome of the pink pigeon with all its chromosomes, and also projected on it are the densities and the positions of the UCEs. So the density of the UCEs are these red little peaks. Now, here we focus in on the chromosome 1, and here the density of chromosome 1. And here we have basically focused in on one UCE. It's about 2000 base pairs. The UCE is that blue bit in the middle, and it has a very high CAT score. And what you can see is that the flanking region, there is lower CAT scores, except this area, which is actually interesting. That's a gene which in fact was not annotated, but it's clearly a gene because we can see there's high positions, high CAT scores for 2/3 of the codons, and there is the kind of the wobble position, the third codon position, the silent position that has a very low CAT score, which is here at the bottom. So this is a typical pattern of a transcribed gene. So you can see that there is high CAT scores for these UCEs, and this can then be used to quantify the genetic load of individuals. Now, there's also other ways to, of course, analyze the genetic load, and this is just a very quick prompt to this software by Swapnil. Vivid, a really fantastic web application that visualizes the impact of mutations on the 3D protein structure. So I think this is a really cool application because what Swapnil has done, he he uses the protein model of a gene, and then he can kind of project on top of that the actual population genetic summary statistics like Tajima's D or DNDS or synonymous, non-synonymous sites. So it's really, really cool application if you want to combine functionality with population genetics. So there's a paper he published or republished in MBE earlier this year. I think if you work with proteins, protein coding genes, and you want to see the population genetics statistics on top of that, this is a really nice application. Now, back to Sam's study, Sam's Peak studies. So what he has done, he has analyzed 6 pink pigeons in the zoo. And this is basically a pilot study to see if he can identify the optimal crosses. You can imagine that in zoos you have, well, you used to use pedigrees, and that's still used to minimize the amount of inbreeding and maximize founder representation and maximize the amount of diversity in your zoo population. But now with genomics, you can also actually look at the genetic load. And this is what Sam is doing here. So basically, he has analyzed those genomes of these 6 individuals, and then he analyzed how compatible are their offspring, their potential offspring. If you would make basically hypothetical crosses, how much load will become realized in their offspring. Now, blue tiles are good crosses with offspring with low load. Red are the bad crosses. These are individuals with a lot of load. Now, this matrix here is the total genetic load of all the pairwise crosses. And this matrix here is the realized load. Now, the realized load is actually that load that becomes expressed, which actually affects the fitness. So there you can see where what the expected fitness would be of the offspring of particular crosses. And of course, you can then use this information to minimize the amount of inbreeding in your next generation. And you can also minimize the population genetic load by choosing the right mate pairs. Now, then another really cool simulations that Hernan Morales has done, where we have tried to see if we can use this information to minimize both the genetic load and still maximize the diversity. So over here in these simulations, on the y-axis, you have genetic variation or genetic load, the bad mutations in red, the neutral mutations in blue, and you have these 3 breeding regimes, the minimizing kinship regime, the minimized load regime, and minimizing both. Now, what you see, if you minimize kinship, basically using pedigree records to avoid matings between related individuals, well, indeed, you maximize diversity. The blue is very nice and high, but also the red bar is high. So there's also a lot of bad mutations maintained in those populations. If you minimize the load, well, indeed, the load becomes very low. That is very nice, but you also lose a lot of genetic variation because basically you select away diversity. Now, with this other regime, whereby you basically minimize both the kinship, so you try to avoid mating between relatives, but you also minimize the load by selecting the best individuals with the lowest genetic load. You can basically reduce the amount of load and still have a high diversity. Now, that is, of course, the kind of the optimal breeding regime in zoos to reduce the amount of inbreeding depression in future generations. Now, in practice, what people have done in the past is trying to minimize the amount of inbreeding in zoos. And basically, if you think of it in a 2D graph where you have genetic load on the y-axis and inbreeding on the x-axis, and you color this in with the extinction probability of a population. So if you have a low load and low inbreeding, well, you're very happy because you have a low amount of chance of going extinct. If you have a high amount of inbreeding and high genetic load, well, you probably will go extinct because you have a lot of inbreeding depression. Now, suggest you're a population that starts somewhere here, so not much inbreeding, but a high genetic load, what typically is for a large outbreeding population. Genetic drift would push down, basically, would push in this direction. You increase the amount of inbreeding. It also reduce the genetic load a little bit because of purging. And that's what we discussed earlier on. There's a little bit of purging opportunity because of some of the load becoming expressed and being removed by selection. Now, what conservation genetics has done for decades is basically just pushing back, trying to minimize the amount of inbreeding using pedigree information, for example, in zoos. Now, with genomics informed management, you can actually both reduce inbreeding and you reduce the genetic load using the similar techniques that Sam has been exploring with his 6 pink pigeons. And of course, that would make the population much more resilient against future genetic drift, because you will end up with a lower genetic load in your population, and therefore inbreeding won't have such a bad effect in the future. So what we then done is we have tried to analyze the effect of this in the pink pigeon using different conservation management plans. So we analyzed genetic rescue, where we would reintroduce pink pigeons from zoos from Jersey and from the UK into Mauritius. We also analyzed demographic rescue and no rescue. And what you can see is that...
Sorry, Cock.
Oh, sorry.
I had to give you a couple of minutes more. I'm really sorry about that.
Sorry. I'm almost at the end. I'm almost at the end. So let me give you a very, very brief. So very briefly, genetic load can be reduced indeed by this kind of selection against these bad mutations, and those regimes are significantly better than just demographic rescue. Now let me zoom to actually the last slide here. So what I think will be really, really cool in the future is the assessment of genetic load in genomes, in reference genomes. And we can analyze this loss of diversity. We can also analyze now the realized load using new techniques, and we can also, I didn't talk much about it, maladaptation and genetic introgression, but there's other things that we can analyze as well using genome data. So let me leave it with that, and I just give you the acknowledgement slides, and then I'll give the floor the opportunity to give questions.